Well, thank you, everybody. It's a full house. It's good to give a talk in full house. I wish my postdoc, Alejandro, was here. It is basically his talk. He took the lead. He made the slides, but he's on J1 from Chile, could not make it. So I am just giving his talk. Of course, we have worked together. These three mentioned over here, we all worked together. Tyler gave a talk in the other data set. This is from the metagenome project. And previous two speakers have given some descriptions of the data, so I will not waste time on that. But I want to make sure that we did not really consider the whole genome. We concentrated on 16S. So the motivation is that based on this NGS data, we wanted to see whether the cities can be differentiated in terms of their OTU profiles. So the data was given as FASTQ files. From there, we found the OTU counts at different taxonomical ranks, that is order, genus, so on and so forth, and use this data under supervised and unsupervised statistical framework to tell a story about the data. And so what we did a bunch of statistical classifiers to predict the provenance of the samples using common taxonomic ranks like order, genus, and family. These three particular ranks, and then we gave tried to do a network analysis to see the difference in the network structures across these three cities. We were eventually we will make a case of finding the network structure of these taxonomical ranks between cities and provide a statistical score, but you can see the visuals of that. And eventually that we will find the differential signature, as I talked about already, across cities based on their common taxonomical ranks. So, okay, so dataset had total samples for these three cities. New York had 1,572 samples, and Boston had 141, and Sacramento had very little, 18 samples. But we had to clean the data. Of course, for cleaning the data, we have used our judgment. Not that that's the best. We have to really, when we write the paper, we have to say why we did that. But however, it's something to be considered carefully. Dataset had the samples taken from wood panels, monitors, screens, etc. We ignored that fact. We did not concentrate on the material as such. So FASTQ files were processed, and then we used the CHIME package for giving the OTU picking, and then the Fred score, the quality score of 35. We chose that. Anything between 30 and 40 is good. So the operational taxonomic unit, the OTU picking for bacterial 16S gene was conducted with this package. I have the reference at the end, and the data was consolidated as counts as three different taxonomic ranks: order, family, and genus. And for microbiome data, as it contains plenty of zeros, you have to do some normalization, some screening to make the data palatable for the analysis, downstream analysis. So we have used this normalization method, which has been determined to be the best one so far. So we use this PGI score, and then we ran the PCA analysis, classification network, and ANOVA. So quickly I will go through this. So for each taxonomical rank, order, family, and genus, only very common levels, not very, just the common levels across the three cities were selected. That is, these particular signals were there for all the cities. So for order, taxonomical rank, we had 19 such common levels, for family 24, and genus 10. PCA plot for considering the three different order, first one order, next family, then genus. You can see that the cities are well separated in terms of their profiles, order, taxonomical ranks, order, family, and genus. Of course, the genus separated it even more. That's what you would expect, because genus is the more specific, at a more specific level downstream. We tried to see that in 3-dimension also, PC1, PC2, PC3, and it looks all right. So the cities, in spite of having this little common OTU ranks, we can still figure out the difference between the cities. Then we tried to classify, really visually, principal component showed us that they could be differentiated, but then we ran the most common classifier like random forest for high dimensional data, and then we report the results for the 3 taxonomic ranks again. For first one, order, and next one is family, third one using genus, and on the x-axis you have the names of the features, and on the you have for the red is Boston, and the next green is New York, and the third one is Sacramento. You can see from the plots that which one is more variable than the other. The classification accuracy on the y-axis it is given, so from there you can see which one is the best. So the next one is that, okay, we have run the random forest classifier, but if it is the best, so what we do for the availability of the classification algorithm, if you have, it doesn't have to be random forest. You can have LDA, linear discrimination analysis, logistics regression. You can have SVM. So whatever it is, given any sets of classifiers that you have, you can construct an ensemble classifier. This is one of our methods research using Baggin and rank aggregation. You can always come up with the best within the set of classifiers that you are using, and we have the package we have the paper reference of the paper, so it is usable anytime. If you have a set of classifiers, you can find one such classifier, ensemble classifier, which will give you the performance which is supposed to be the best one, close to the best one within the set. So I'm not going through the algorithm. I don't have time for that, but this is the data. Then next is that I will report the results of all the classifiers for order, family, and genus again, and the 3 different classifiers, that random forest, SVM, and the ensemble. We used in SVM, random forest, and then the ensemble classifier, and we reported the sensitivity, specificity, all the common stuff that you do, AUC, for accuracy, all that. You can read the plot and see that most of the time the ensemble is doing its job. It's the best given the data. And although it's really hard to read from here, the colors are too light, I understand that. New York was little hard. For x-axis, these plots are given to you for different cities. The Boston versus New York, the classification of Boston versus New York, and then Boston versus Sacramento. The next one is Boston versus Sacramento, and the accuracies are plotted on the y-axis, and different colors are for different classifiers. What happened, Boston versus New York was little hard to classify. they were a little similar as far as order is concerned, the taxonomic rank order is concerned. Even for the family, that was the case for genus. So it looked like New York and Boston, it's hard to classify, but others are all right. For all the classifiers, Ensemble was and Random Forest were performance-wise, they were mostly the same. So then we have considered the network structure. So the story is that yes, we can classify based on this small number of features. Also, we can classify the cities. It's important, but imagine a situation of endemics. If you have the profiles of different cities, bacterial profiles of different cities, you can quickly address that which are the cities that we should go and take care of it with some treatment on all. So I think differentiating different cities with their microbial status is an important problem to consider. So next, we have tried to see that whether we can construct networks based on these taxonomical ranks. And we have done it. We have done it with partial least square regression. We have our previous work constructing network with reverse engineering of network with the package called DNA. Actually, it does even more. This is the package where we describe the paper where we have done the methods for constructing networks for genes using partial least square regression. But here we have constructed it. There is a package called DNA package differential network analysis. It not only constructs the network, but it can figure out the difference of network structures between different groups of disease. Here, the different cities. That was done in the context of microarray data, but it can be extended to next generation sequencing data. So this one, only thing is the modeling will be different. It's a count data. We have already extended it in the context of network construction for next generation sequencing data with negative binomial model. But you have to do the dimension reduction anyways. So anyway, the story is that Sacramento looks really, in terms of network, Sacramento looks very different from the other two. Looks like it is centered around one such order, but here the New York and Boston, they have multiple hubs, and also it's centered around this. And the bacteria that the rank order across all the cities reveal the common class, that alpha proteobacteria and gamma proteobacteria. They are the central nodes between Boston and New York. So these round ones, the round that we have done over here, they all fall into proteobacteria, what the previous speaker also said. So data is telling us something. Next is that we have tried to run ANOVA also to see the statistical difference between the different cities. The most common intriguing result is that all the cities, they are really different in terms of ANOVA, doing ANOVA, and they are explained in this 14 profiles. For all these 14 taxonomic ranks, they are really different. And for other counts also you can see little bit of difference, but mostly, so A, B, C rank, they are, if they are similar, all of them will be A or B, 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 whatever. So you can read from there. So what's the result? Bottom line is the cities are really different, and Boston and New York, they are a little similar in terms of these profiles, but Sacramento is very different. So this is the alpha diversity or whatever, that's for different cities we have plotted. And the colors are for different orders, order, genus, and family. You can see that Sacramento diversity is much higher. New York is probably at the bottom. So it doesn't have enough diversity. So the conclusions: bacterial populations can be city-specific and their signal is useful to predict samples' provenance. PCA analysis showed that 16S profiling data contains strong enough signal to cluster samples from different cities. Additionally, PCA showed high proportion of variability explained by 3 components of all taxonomical ranks. And predicting provenance based on bacterial metagenomic composition, that was highly effective for all classifiers tested. Order was the best, of course. And then the next is ANOVA also showed that the cities are very different. Network analysis showed that network structures that reflect the proximity of Boston and New York, because Boston and New York look like they are non-centralized networks, but Sacramento had a central node. So it's in West Coast, so maybe it is different. And bacterial composition found in DNA sequencing can be used in the context of statistical framework to assess differential patterns of diversity, which can be influential in forensic ecology and other sciences as well. And that's our message. And please do get back if you have any question regarding this.
Thank you, Susmita. Well, the questions will be at the poster session.
Huh?
So questions will be during the